Welcome, folks, to microbiology. Today we're going to be covering Chapter 3 on cellular structure and function. And although we will be focusing on prokaryotes, this eukaryotic cartoon was fun for our discussion today. And so in our discussion today, we're going to be looking at the characteristics of life and how each cell, no matter which domain it's from, actually has all of those characteristics of life. Then we're going to really sit down with the bacterial cell and look at its structure and function. And then we're going to conclude our discussion today with a comparison between bacteria and archaea. We will not focus on eukaryotes, although they are covered in this chapter, and I do welcome you to look over that content. Okay. What we'll do is move in subsequent chapters to even more detailed discussions of metabolic functions and genetic information. And all of these discussions are really built on a foundational understanding of macromolecules within living systems. So I definitely recommend that you review that content, which I'm sure you covered in prerequisite classes to this. Actually, the first link in the description below is a video review of macromolecules. Okay. Also, I recommend that you review previous content to this chapter and review chapter 3 or preview chapter 3 itself before we jump into the content. So feel free to pause the video, prepare yourself for this lecture, and then jump back in. Okay, so what is life? In our discussion in chapter 1, we talked about the history of microbiology, and we saw that over centuries, scientists and microbiologists really worked to understand what is a living system, what is disease, what are the components of living systems, what are cells, how do they function, so on and so forth. And so, historically, as we were still grappling with some of these ideas, there was this understanding that maybe living systems were actually at an elemental level different from non-living systems. They were actually composed of different components. And of course, what we know now is that living or non-living, all systems are built upon the same common elements within, our Earth and our systems. But living systems tend to be carbon-based in their forms versus non-living systems, which may be more diverse. And so that understanding really, there was like this pivotal moment in 1828 where a researcher named Frederick Waller was actually able to develop an organic compound in the lab using various elements, and that compound was urea, where it became obvious that even organic compounds are made of the same components of non-living systems, but just, like we said, more carbon-based. And so what is a living system? If it's made up of kind of the same components as non-living matter, how do we really say, okay, this system is living and this one is non-living, right? And I'm sure you've been through these characteristics in other classes, and what your textbook focuses on is growth, reproduction, response to stimuli, and metabolism. Of course, there are a couple other things, and I actually prefer a slightly more expanded list where we say that a living system is able to acquire and use materials and energy. It's actively able to maintain organized complexity. We're going to talk more about that in upcoming chapters. It's able to sense and respond to stimuli in its environment. It's able to grow and reproduce, and really essentially, the systems are able to evolve collectively. So an individual organism will not evolve, but a population of organisms will evolve, or a community of organisms will evolve. And so when we go through this list and we look at, a system that in nature, and we look at these specific characteristics, it allows us to identify whether that system is living or not, right? And so at a macro scale, sometimes it's pretty intuitive when we look at an animal or a plant or something like that. We can say, okay, this one seems to be a living organism versus a non-living piece of debris or component in the environment. But at these smaller levels, when we're looking at one individual prokaryotic cell, for example, it can be a little bit more difficult to exactly identify, each of these characteristics within. And really the primary example of that is viruses, right? And we won't talk about viruses so much in this chapter, but in chapter 13, we're going to focus on viruses and also prions. And so when we look at a virus and we go through these characteristics, we can say, well, how are they actually qualifying in each of these characteristics? And do they actually qualify as living systems? And viruses are really great examples because they do have kind of living characteristics, but as a whole, they tend to be considered non-living microbial agents or particles. And so I'm going to recommend that you pause the video here and do a little bit of background research, maybe jump into chapter 13 for a minute and look at viruses and how they can be kind of at that boundary point between living and non-living systems. And actually, our first component of our discussion post is you're going to actually take viruses as an example and go through this list of living systems that they can acquire and use material as an energy. They actively maintain organized complexity. They sense and respond to stimuli. They grow, they reproduce, and they evolve collectively. And consider those different components and how viruses do or don't actually kind of check off those various characteristics. So go ahead and pause the video here, start that part of your discussion post. There'll be a second part of the discussion post later in the video, and then you can conclude that and post it at the end of our lecture today. Okay, welcome back. So once we've identified that a system is indeed living, what we'll see is that there's so much diversity, right? Just these images that you see here, there's so much diversity in what a living system actually is. And so from a biological perspective, being able to then categorize living systems and understand how they relate to one another is really essential. And so we use what's called the Linnaean classification system to understand how organisms are categorized and related to one another. That system is based on similar characteristics, but also really, really importantly, it's based on genetic phylogeny, okay? And so you can essentially see here 3 different microorganisms. One is a eukaryote, one is an archaea, and one is a bacteria. And you can see that we essentially, within the Linnaean classification system, we look at each of these organisms in all other living systems within context, okay? So here you see all organisms, right? All organisms. And then we start to look at them in more and more similar groups. So as you know, we have 3 domains of life: eukaryotes, bacteria, archaea, and these are distinct groups of organisms that have within them similar characteristics, but they also have a significant amount of diversity. And so within eukarya, for example, we have a number of kingdoms, and baker's yeast falls under fungi. And then we get more and more specific. So different phyla of fungi, different classes, different orders, different families, and then we get to the actual genus and species, okay? And so here we're looking at the genus Saccharomyces and the species Cerevisiae. And so now at this point, when we get to the genus and species, we're looking at one species of organisms that's actually similar enough that it can reproduce successfully within, and we can identify that species based on actually the genus and species names. And that's actually how we name organisms in biology, is we take their genus and their species, we combine them together, and that's the name of the organism. We format that in a very specific way. When we're handwriting out the scientific name of an organism, we always underline it. And when we're typing it, we always italicize it. The first letter of the genus is always capitalized and everything else is lowercase. And just like with abbreviations, the first time you mention the scientific name in a paper or a chapter, you'll write it out in full: genus, species. And then thereafter, you can actually abbreviate the genus to just the first letter of the genus name, and then add the species name to that, okay? And so this will be important for anything that you submit in this class, lecture, or lab as well. So keep that in mind. Okay. And then we can do the same with these different organisms as well. So E. coli is one species, but it is part of the domain bacteria. And we can go through and categorize organisms into more and more similar categories until we get to the one individual species, which is E. coli. Okay. And so within this system, we're able to look at both specific individual species, but also how those species are related within the broad context of living systems throughout the world. Okay. Another thing that this, specifically the naming standard allows us to do is speak the same language, no matter which scientist around the world, no matter which language they speak, we all identify these organisms based on their scientific name. And that means that we can go back and really through the scientific literature, keep track of what is known about each individual species. Okay. There's a fun mnemonic that I was taught by my biology instructor that allows me to keep track of these levels of hierarchy. And that is dopey kings play chess on funny green squares. Dopey kings play chess on funny green squares. And so that is domain, kingdom, phylum, class, order, family, genus, species. You might know other mnemonics that help you keep track of that, but that is kind of a fun one. Okay, so if we then take these groups of organisms, right, eukarya, archaea, bacteria, and we look at them using some of the tools that we had discussed in chapter 1, like the microscope, this is specifically an electron microscope, then we can see their individual cells, right? If we're looking at domain eukarya, we have organisms that are both multicellular and unicellular. So a unicellular example is this paramecium right here, which has this beautiful, just external 3D coverage of cilia. Cilia are kind of these little hair-like structures that allow for the movement of this organism. And those cells tend to be larger. So if you see this kind of scale right here, this is 40 micrometers versus this scale here, which is 1 micrometer, larger than the cells in bacteria and archaea. And there are a couple of exceptions to that, which we'll talk about as well. But, just generally a rule of thumb is that kind of more compartmentalized organized structure of eukaryotic cells allows them to kind of have a slightly increased size or an increased size compared to bacteria and archaea. Whereas here you see these bacteria, they have kind of simple individual single cells, and all bacteria and archaea are single cellular organisms. They may form colonies, but we don't see multicellular structures or organisms or organ systems or organs that arise from bacteria and archaea. There are some really fun colonial bacteria that we will talk about specifically in chapter 11. So look forward to that as well. Okay. So in terms of these bacterial cells, they're typically around 1 micrometer in size, although there are exceptions. There are smaller bacteria, larger bacteria, specifically bacteria that are obligate parasites. They tend to not have to have a larger size, so they can be really small, and a couple of really interesting examples of larger bacteria as well, which we'll talk about further in the class. And that's compared to these eukaryotic cells, which are more in the range of like 10 to 100 micrometers in diameter instead. The eukaryotic cells are the ones that we had talked about in chapter 1, which are algae, protozoa, fungi, animals, and plants. Whereas our prokaryotic cells are just the bacteria, or not just, because these are large, diverse domains and groups, but bacteria and archaea. So just a quick reminder that those eukaryotic cells tend to be more complex and compartmentalized. And so you have specific organelles within the cell, or small compartments within the cell with specific tasks. So here you have your nucleus, for example, your endoplasmic reticulum, your Golgi apparatus, and all of this is compartmentalized in terms of their function. You also have some kind of motility structures like cilia and things like that as well. Whereas our typical prokaryotic cell is really, really simple, really simple. So it's a single cell, and it has a region where there's genetic information, because genetic information is essential. And then within its cytoplasm, it has the proteins that it needs to kind of implement that genetic information, like ribosomes. And then it might have some storage components like inclusions. And that's about it that's going on inside of the cell. Now, one thing about bacteria and archaea, since they're individual unicellular organisms, they do tend to need to have a significant amount of protection outside of their cells. And so what we're going to do in this lecture is we're going to start from the outside and go in. And as we do that, you're going to see that there's layers of protection for these cells, which makes sense because they're unicellular organisms within a relatively harsh environment. And so those layers of protection allow them to maintain their internal metabolic functions separate from the external world. Okay. So let's talk more about prokaryotes. We had mentioned that prokaryotes generally lack membrane-bound compartments, whereas eukaryotes have those. And so although they sometimes have membranes that concentrate different pigments or molecules on them, they won't have specific kind of compartments that are membrane-bound. And we had mentioned that they're small in diameter, and they include bacteria and archaea. Okay. So a little bit of a size discussion here. If we want to look at the size of these organisms from various domains, technically the largest cell is actually a bird's egg, because that's one individual egg. But if we look at a more characteristic individual cell with, its own kind of small membrane, plasma membrane, a human egg is pretty large as well for just individual cells, microscopic cells. The human egg is about the size of the period at the end of a sentence when you're writing in 12-point font times New Roman. So just visible with the naked eye, okay? Also, you have some parasitic eukaryotes, like this Giardia, which are just large enough that they're going to be visible with the naked eye. Whereas bacteria are many, many times smaller than that. So you can see here 1 micrometer compared to that 14 micrometers of the Giardia. And then viruses will be many times smaller than that. Of course, within bacteria, you have some exceptions. So we said, for example, Mycoplasma is an obligate parasite. And this is actually the organism that causes tuberculosis. And it's between 0.2 and 0.8 micrometers. And that's basically because it relies on its host. It doesn't really need that large size. It also has a smaller genome because it relies on its host. So it's kind of one of the smallest free-living bacteria that we know of. Whereas Epilopiscium, which we'll continue to discuss throughout the class, is very large, about 200 to 700 micrometers in size, and it has some really unique features. When we get to chapter 11, we're going to talk about those. You can basically almost see it with your naked eye. So kind of a very large bacteria, the largest bacteria that we know of. Okay, so what we want to do now is continue to focus on bacteria specifically, and we're going to look at a bacterial cell and the components of a bacterial cell, and really dissect those. And then at the end of the discussion today, we're going to talk about how those can be different from archaea. So first, we said we're going to start on the outside of the cell and move in. And we said there are a significant number of protective layers on the outside of a cell. So this is what we call a glycocalyx. Okay, this is another type of glycocalyx, and this is very common in bacteria, and it's essentially a sugar coating. Glyco glyco is always in reference to sugars, like glucose, for example. So it's a sugar coating that's around the cell. You have two types of these. You have either a very kind of structured, organized, what we call capsule, or you have what just kind of looks like a snot mess, which is called a slime layer. So this just sticky jumble of sugars that are around the cell. Both of them do the same function, which is to protect the cell from the environment, specifically from desiccation. Okay, and so those two different types of capsules, of glycocalyces, one is the capsule and one is the slime layer. Okay. And so that those tend to just be on the outside of the cell and are able to protect those organisms from their environment. Okay. Oops. Another external function structure is the flagellum, which allows for the movement of cells. Okay. And so let's get a picture of a flagella here. Okay. There are different kind of numbers and arrangements of flagella, but these structures that come out of the cell and allow for that cell to move, those are what we call flagella. Okay. They are not present in all bacteria. Some bacteria are motile and some aren't, but when they are present, they hook into the cell membrane and the cell wall of that bacteria and allow for motility. So there's a couple of components of the flagella that will cover, the flagellum that will cover as the filament, the hook, and the basal body. And these look slightly different depending on the way that the cell wall is organized. And we're going to talk a little bit more about the cell wall later in this lecture. So come back to this idea of the flagella once we get there. Okay, so you can see here, you have this hook here, which is a protein that actually connects the flagellum to the cell wall of the organism, and the filament, which will extend out. So this is kind of your cell as a whole, and this is a cross-section of where that flagellum meets the cell right here. And so you have that hook protein that allows it to kind of hook into the membrane, and then you have the filament, which is connected on this side. Now, what the hook protein is attached to is a basal body, and the basal body is connected within the membranes and cell wall of the organism. So we're going to talk a little bit more about some bacteria are what we call gram-positive, and some bacteria are what we call gram-negative. And you can actually already see the distinction here. So the gram-positive organisms have a thick cell wall, whereas gram-negative organisms have a thin cell wall, which they actually augment by having a secondary cell membrane. So for a gram-positive organism, you have the traditional cell membrane and then a thick cell wall. And the way that that basal body connects is using these two protein rings that are connected at the cell membrane level. Whereas for gram-negatives that have the cell membrane, but then also a thin cell wall and then another outer membrane, you actually have 4 of those protein rings that hold that flagellum in place. Okay. And you can kind of see that here as well. And I will let you watch this video.
The long whip-like portion of the flagellum is called the filament. Filaments are composed of many identical globular protein molecules called flagellin. Molecules of flagellin form helical chains around a hollow core. Certain pathogenic bacteria can be identified and classified by differences in their flagellar proteins. In contrast with eukaryotic flagella, no membrane covers the filament of a prokaryotic flagellum. The whip-like filament inserts into a curved structure called a hook. The hook is composed of a different protein and connects the filament to the basal body. The basal body is composed of different proteins than the filament and hook. The basal body is composed of a rod and a series of either 2 or 4 rings, which anchor the flagellum to the cell wall and allow the flagellum to rotate 360 degrees. You're viewing a flagellum from a gram-negative cell. A gram-positive cell would only have 2 rings in its basal body, both of which are attached to the plasma membrane.
So, um, so as we saw with that video there, you're actually looking at kind of a 360-degree motion of flagellum. I think sometimes we think of flagella as tails, and we think of the motion as kind of like a snake-like back and forth motion, but actually flagella are moving at a 360 degrees. And the way that flagella are organized, either they could be at a terminal, or they could cover the whole 3D surface of the organism, or we have some really interesting organisms called spirochetes that have some really unique arrangements. So I'm gonna also show you some of those.
Cells with a single flagellum are monotrichous. When the flagellum rotates counterclockwise, the bacterium runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Amphitrichous bacteria have a flagellum at either end of the cell. Bacteria with a tuft of flagella at one end are called lophotrichous cells. When the flagella rotate counterclockwise, the flagella bunch up and the cell runs. When the flagella rotate clockwise, they separate and the cell tumbles. Paratrichous bacteria have flagella covering the surface of the cell. Runs occur when all of the flagella rotate counterclockwise and become bundled, while tumbles occur when the flagella rotate clockwise and separate.
Okay. And here are spirochetes, which have what we call an endoflagellum or endoflagella that actually wrap into and around their cells. Oh,
Disease is caused by.
1 second. There we go.
Spirochetes are a group of bacteria that are spiral-shaped. They move in a corkscrew-like fashion, using flagella that do not protrude from the cell wall like typical flagella. Lyme disease is caused by a spirochete called Borrelia burgdorferi. Another spirochete, Treptonema pallidum, causes syphilis. Spirochetes have an axial filament that lies between the outer membrane and the cell membrane. The axial filament winds around the outer edge of the cell. The axial filament is made up of a bundle of modified flagella called endoflagella. Rotation of the endoflagella causes the axial filament to rotate around the bacterium, which causes the spirochete to corkscrew through its medium. It is thought that this manner of movement allows the spirochetes to move more easily through viscous human tissues and fluids, such as mucus. Then they can easily attach to the cells below.
So some really interesting kind of anatomical features of flagella, and also how those flagella allow for those organisms to kind of function. And specifically with spirochetes, we're going to talk more about spirochetes and some of the illnesses and diseases that they cause. But that corkscrew motion is very unique and interesting. You also saw in the video the mechanism of motility is called run and tumble, which I always think is really fun, because essentially the way that we as individuals respond to our stimuli as human beings, for example, is we see, hear, touch, feel, sense something in our environment. If that is a positive attractant, then we will move towards it. If it's a negative or repellent, we will move away from it. Bacteria do the same thing, but in this kind of strange, random way. Not completely random, but random in some ways. So they actually run, so as you saw, that counterclockwise motion that allows them to move forward. And if they sense that they're getting closer to an attractant, are farther from a repellent based on the chemical signals in their environment, they'll continue in that direction. But if they sense that they're getting closer to a repellent or further from an attractant, based on those chemical signals, they'll actually tumble, which will reorient them randomly. And then they'll run again. And then they'll sense their environment based on the chemical stimuli. And then they'll either tumble if they need to move in a different direction or continue in that direction, right? So it's very interesting that they're kind of moving, sensing their environment, taking in their stimuli, and then responding by tumbling, which kind of reorients them randomly. And then they continue until they are able to get to where they need to go, right? Either towards a nutrient or something like that, that's an attractant, or away from a repellent. So just because it's kind of fun to see this, here is a video of the run and tumble looking at various types of arrangements of flagella.
The motility is the ability of an organism to move by itself. The motility of a cell may enable it to move toward a desired environment or to flee from a harmful environment. Bacterial cells use structures called flagella, or in the case of spirochetes, flagella-like structures called axial filaments to enable this movement.
So you can see here how you have this kind of run and tumble mechanism where organisms are moving directionally, and then they can randomly change direction and then see if they can kind of get to their destination. Even though we have this really interesting and unique mechanism for motility, bacteria can actually move really quickly. So it's kind of a moment to take pause and think about our perspective, actually. And think about, if we think about living systems, we often think as the fastest living system on land being the cheetah, or the fastest in the air being the falcon. But really reorienting ourselves, because some of these bacteria are able to reach speeds that are almost 10 times as fast as a cheetah, even though they're doing this kind of funny run and tumble mechanism to get where they're going. And so they have these really high speeds where they can move about 60 cell lengths per second. In that same thread, although this is a eukaryotic example, it reminded me that actually some of the top speeds in living systems are actually outside of the animal world. And so I thought it would be fun to bring that in. So kind of just mental break that we're moving into a eukaryotic example, and then we'll come back to bacteria in a minute. But like we said, oftentimes we think of the fastest, even eukaryote generally, as maybe being a cheetah or a falcon, but really the top acceleration, the top speeds for acceleration in the eukaryotic world are held by a fungus and a plant. And so you might be thinking like, what? Aren't fungi and plants basically stationary? And so what it is the release of spores, fungal spores, and in the case of the plant, a specific type of dogwood spore is so quick that it actually holds the record for the fastest acceleration. And so I wanted to show you that here. This is actually a coprophilic fungus, which means that it grows on dung. And so these researchers are actually literally watching a piece of dung and waiting to be able to film the release of those spores, which, like we said, is a record holder. And it's so fast that if you just look at it with your naked eye, you'll just kind of see the spore disappear. Oh, and he touches his face with his glove. Sorry, this is so random, but he touches his face with his glove, even though he's working with dung. Anyways, but it's so fast that you'll actually just you'll just see the spore disappear. But when it's slowed down, you can actually see how quickly that spore is released into the environment. So this was a BBC special on this Pilobolus fungus, which was really fun. That is in the course resources if you'd like to see the whole video. But so just to pause to think about our perspective a little bit, to think about what we know in the macro world and what exists in the micro world, but oftentimes we don't detect or we can't see it. So keeping ourselves aware and our minds open as we're moving through this micro world. Okay. Okay. So what we've talked about so far, and we're going to move back into our discussion of bacteria, actually, and away from this fun eukaryotic example where we gained a little bit of perspective. So we had just finished talking about motility in bacteria. And so they're able to move with these different arrangements of flagella, either outside or endoflagella, if we're talking about spirochetes, of their cell. And that allows them essentially to move towards attractants and away from repellents. We also talked about how bacteria and archaea, well, when we talk about archaea, it would be the same for flagella, but stay tuned. These are unicellular organisms that need to have protective layers within their environment. So they have these sticky layers that they kind of keep around them called glycocalyces. You could have a glycocalyx that is a capsule, and so it's kind of this organized kind of standard structure of those sugars that gives you this organized and usually a little bit tighter of what we call a capsule. Or you can just kind of have this mess of sticky sugars called a slime layer, and both of those protect the cell. So let's continue with our discussion of anatomy. And what we're going to see here is a couple of other structures that extrude from some bacterial cells, not all, and it's pretty much characteristic, like how flagella are arranged, whether a capsule is made, whether a slime layer is made, whether you have some of these structures that we're going to talk about in a minute, tends to be characteristic to specific species, okay? But we're going to continue to discuss some of these outer structures that allow for the bacteria to kind of exist within and interact with its environment. And so our next discussion is on what we call Fimbriae and pili. And these are both structures that come out of the cell. So here you're looking at a distinction between Fimbriae, which are these long protein structures that come out of the cell, versus flagella, which are even longer and are for motility. Fimbriae, as you can kind of imagine from this image here, are more for attachment and interaction with other cells, whereas flagella are actually for motion, which we had talked about a minute ago. And then pili, just so we get a distinction of the three different structures, pili are actually these kind of these anatomical structures that come outside of the cell and attach to another cell, and they're specifically for transfer of genetic information. So not motility and not really attachment, but for transfer of genetic information, a plasmid, which is a small piece of DNA, is transferred into the next cell. And then that pilus is then terminated. And so here you're looking at the distinction between Fimbriae, which are these kind of spike-like proteins on the outside of the cell that allow for it to attach to a surface or other cells, and then a pilus, which is connecting to the next cell. So we have flagella for motion, Fimbriae for attachment, and pili for exchange of genetic information. Okay, so let's look at those Fimbriae and pili in a little bit more detail. Okay. And so Fimbriae are these sticky bristle-like projections that are coming out from the cell. They are shorter than flagella, and they are not moving like flagella. And they allow for the cells to adhere to their environment and to other cells, usually both the environment and the other cells. And that's one of the primary functions, is to produce biofilms, right? And so basically, a lot of what we do in our cleaning world is we're trying to clean biofilms off of surfaces. So maybe you're in a medical lab and you're having to, do various types of microbial control on surfaces and tools to make sure that biofilms are those sticky surfaces of mats or masses of microorganisms don't actually accumulate on your tools or surfaces in a medical setting, right? And so biofilms are really important for bacteria because it allows them to aggregate where there are resources, right? Maybe somebody has some sort of implant in their body, and then those organisms, bacteria, are able to aggregate on that implant because the inside of a human body is full of resources and full of, the temperature and metabolites and so on and so forth, right? And then we have many different techniques to try to make sure that that doesn't happen. Or just even in the kitchen or the restroom, sometimes we have biofilms that aggregate like in moist surfaces like a bathtub or a sink, or a sponge, which we will visit in lab, because that sponge is full of nutrients, and those microorganisms are able to aggregate together and on the sponge so that they can actually utilize those nutrients. From a medical perspective, from a food safety perspective, biofilms can be problematic oftentimes. And so Fimbriae are really some of the essential components of the cell that actually allow for the formation of biofilms. Okay. Those glycocalyces can actually be helpful in that as well. And so here you can actually see a biofilm where there's an aggregate, in this case, of different types of bacteria that are connected to one another via their Fimbriae and their glycocalyces that allow them to kind of aggregate together. And we're going to continue to talk about biofilms and various interactions between microorganisms as we move through the course. Okay. Then we said we have this other structure, which is basically this long connective structure that allows for the exchange of genetic information. And so we're going to talk about this more today, but also in some of our subsequent chapters, where a bacterial cell or a prokaryotic cell will have its own full genome within a chromosome, but sometimes it'll have these non-essential genes within what we call plasmids. And these are just circular pieces of DNA that are very small that move within the cell and within the cytoplasm of the cell. We can actually use those plasmids for lab purposes, and we will do that actually in our lab. We will kind of utilize some artificial plasmids that we've made. And those plasmids, not the whole genome, not the whole chromosome, but those small plasmids can actually be exchanged between organisms. And so pili allow for that. So here's a single pilus that's connecting these two organisms, and there's a transfer of information. Here's another electron microscope image of a pilus here. Okay. And so we have those external structures, flagella, which are for motion, Fimbriae, which are for interaction with the environment and other cells, and pili, which are for the transfer of small pieces of genetic information. And we call that horizontal gene transfer because it's transfer of genetic information between pairs. Just a note on horizontal gene transfer and the specific type of horizontal gene transfer in this case is called conjugation. Remember that bacteria, archaea, any prokaryotes, they do not reproduce sexually. So it's not actual reproduction, it's just a sharing of genetic information. And this is oftentimes how we get antibiotic resistance spreading through a population or a community, is through these plasmids that are transferred peer to peer. But it's not sexual reproduction in that these two are not parent cells that will produce a daughter cell. They're just two peers that are sharing information. Okay, so let's continue to move through the anatomy of a bacterial cell. And so the next structure that we'll look at is the cell wall. And so the cell wall is another, so we know that, all cells from previous classes have a cell membrane that separate it from the environment. Within eukaryotes, sometimes we just have a cell membrane, and sometimes, like with plants and fungi, there's also a cell wall that protects that cell. Within prokaryotes, all cells need a cell wall because they need some protective layer, again, to protect their plasma membrane. Well, almost all. There are a couple of obligate parasites that have lost their cell wall. Almost all prokaryotes have cell walls, though. And so that cell wall is a structure on top of the cell membrane that allows that protective layer. These cell walls are also really important because they essentially give the shape of that organism. So when we talked about cocci, bacilli, and spirals, essentially that cell wall is what's dictating the shape. And in those organisms, those couple of organisms that don't have cell walls, they actually are pleomorphic. They just kind of have whatever shape they want to have. Also, the cell wall tends to be one of the primary targets of antibiotics and antibiotic drugs. When we get to chapter 10 later on in the term, we're going to talk specifically about how antibiotics target different processes within cells. And one of the primary mechanisms is targeting that cell wall. And that cell wall has a different component from any eukaryotic cell walls. And that's one of the ways that it allows for direct targeting of bacteria or prokaryotes. Although there is some issue because we have so many bacteria and archaea even in our microbiome. And so that can be an issue with antibiotics sometimes. And we'll talk more about that throughout the term. Okay. And so that cell wall giving the structure of the cell. And as we mentioned before, the way in which that cell and also the cell wall divide then gives rise to the arrangement of those groups of cells. Okay. Another really essential characteristic of cell walls is that there tend to be two types. Now, there are two predominant types, and there are a couple other exceptions that are unique, and we'll talk a little bit about those too. But the two dominant types of cell walls are either gram-positives or gram-negatives, which we mentioned briefly when we talked about flagellar attachment. Whether an organism is gram-positive or gram-negative, they have a cell wall made up of small sugar components that are organized together. If it's a gram-positive organism, that cell wall is very thick. If it's a gram-negative organism, that component of the cell wall that has those arranged sugars is thin, but it does actually have an additional layer on top, which we'll get to protect that thin cell wall. So let's look at that sugar component of the cell wall. Okay, the components, the sugar components, or that kind of aggregate of sugars is called a peptidoglycan cell wall, and it's made up of just alternating NAG and NAM sugar components. Okay, and I'm going to tell you more about NAG and NAM and those sugars. They look similar to glucose. Okay, so glucose is a sugar that makes up the cell wall in plants, for example. And so the cell wall in plants is just an arrangement, cellulose, the stuff that gets stuck in our teeth, right? And that's why it's so good for making a cell wall, because it's a protective layer. It's just an arrangement of these sugar components of glucose. Well, NAG is N-acetylglucosamine, and it has a very similar ring-like structure, but then it has slightly different kind of chemical attachments to that ring. And then NAM is N-acetylmuramic acid, which again looks similar in its structure. So all of these points are all carbon attachments, and then you can see what are attached to them here. And so NAM, again, similar to glucose. And then these two subunits are connected to each other, a NAG to a NAM. And then those are connected together to form this whole series, which you see here, of sugar components that are all connected to one another to form a cell wall. Very similar, again, to plants, where we have chains of glucose that are connected to one another to form the cell wall of the plant. Okay, so even though this is a eukaryotic example, it allows us to kind of think about the cell wall within a structure that we're already familiar with from prereq classes. Okay, and so let's go back to the prokaryotic peptidoglycan layer, which again we said is similar to the cell wall of a plant, for example, in terms of it having these sugar subunits, but they're different subunits. So we have those alternating sugar subunits. Also, the subunits are connected by cross bridges. You have a tetrapeptide cross bridge, which is tetra meaning 4, peptide meaning protein. So you have these 4 proteins that connect your NAM subunits to one another, and they connect vertically. You also have horizontal subunits that are connected, and these are chains of amino acids that connect vertically. And so all of those sugar components in the peptidoglycan layer, with all of the cross bridges together, make a very, very hardy cell wall that allows for the organism to be protected from the external environment. Okay. There we go. Now, we said that there's two different kind of predominant categories of, I want to show you a comparative image. Let me grab that image really quick. Okay, I'm just gonna hide part of this image because we're gonna get back to it. But a gram-positive organism is going to have the cell membrane, which covers the whole cell, which we haven't talked about yet, but it's similar to cell membrane, the function of cell membranes in other cells that you're aware of. And so the whole 3D structure of the cell, so if this is a cell right here, the whole 3D structure of that cell is going to be covered in the cell membrane, and then it's going to be covered by a thick peptidoglycan cell wall. And then here you can see your NAG and NAMS subunit components here. Okay, so that's gram-positive. Whereas gram-negative is going to have the cell membrane, as all cells do. And then it's going to have a thin peptidoglycan cell wall. But to compensate for that thin peptidoglycan cell wall, it also has this really unique outer membrane. And actually, that outer membrane has some really unique characteristics that are related to pathology. So we're going to talk about these two different types of cells next. Now I'm going to unhide part of what you didn't see in this image. We're also going to talk about a couple of unique exceptions to these anatomical structures and features after we talk about gram-positives and gram-negatives. Okay. So first, we're going to talk about gram-positive bacterial cell walls, and those are the organisms that have a thick peptidoglycan cell wall above or covering their cell membrane. And so we know that they have that thick peptidoglycan kind of components. They have a couple of other things. They have a couple of acids that anchor the cell wall to the cell membrane and anchor within the cell wall as well. And those are called teichoic acids. And then lipoteichoic acids have just a little lipid attached to them that allow them to actually connect to the cell membrane itself. And you'll see that in a second. And so that really gives a really thick, really nice boundary between the cell membrane and the external environment. So here you have a gram-positive cell. Here you're looking at the membrane and the cell wall. And here we have a close-up of the cell membrane down here with a thick peptidoglycan cell layer, teichoic acids that are stabilizing that cell wall, and then lipoteichoic acids that are actually anchoring the cell wall into the cell membrane. Okay. We have a couple of really unique organisms called mycolic that have mycolic acid in their cell wall, and we're going to talk about those in a minute. And those are really interesting, and they do cause pathology. But I first want to talk about gram-negatives first. Okay, so gram-negatives are the organisms that we said they have a cell membrane, and then on top of that they have a thin peptidoglycan cell wall, and then they have another membrane on top, which is called the lipopolysaccharide, or has within it lipopolysaccharides and other proteins that make it really unique. And so let's look at that. So here you're looking at a gram-negative organism. You're looking at the cell membrane here in blue and the cell wall in this tan color, and then we're enlarging that so that we could look at it a little bit more clearly. So here you have your cell membrane, a thin peptidoglycan cell wall, and then this outer membrane that has all of these lipopolysaccharides on it. So you can see these that are extruding from there. Your body actually recognizes lipopolysaccharides, and specifically a protein called protein A that's within that lipid, or sorry, a lipid called lipid A, which is in that lipopolysaccharide. And it can react, and some of those reactions from your immune system, inflammation, things like that, are actually part of the cause of disease with these organisms. And we're going to talk more when we get to chapter 11 about lipid A and lipopolysaccharides and things like that within gram-negative organisms. But for now, just know that this is what your gram-negative organism looks like in terms of its cell wall versus a gram-positive, which has a thicker cell wall. Now, this is really essential because it allows us to categorize organisms, and it allows for a visual way for us to see which organisms we have in the lab as well. And so one of the first techniques you'll do in lab is what's called a Gram stain. And so a researcher named Gram actually came up with a way of distinguishing these organisms by, instead of using one stain in the lab, he uses a series of steps where you have a stain, a rinse, and then a counter stain that allows you to actually see the difference between these organisms. So what will happen is a gram-negative will show up pink in this process, because essentially the primary stain that's used in the process will be washed out because that thin cell wall won't hold it. Whereas if we go back to gram-positives, these gram-positives will always stain purple in this gram stain, because the primary stain, which is crystal violet, will actually be held within, because of the process that we use, will be held within the cell. And so we're going to talk more about that in lab. You're going to look at what is the primary stain, what is the mordant, what is the rinse, and what is the secondary stain that are all used in that procedure, so that we can visually see under the microscope whether we have a gram-positive organism or a gram-negative organism. Okay. So I promised you that we're going to talk about a couple of unique organisms in relation to their cell wall. So you have just a few organisms, specifically mycoplasmas, and some of these cause a milder form of pneumonia that actually don't have cell walls. And these are very unique and very rare. And the reason why they don't need a cell wall is because they are parasites, so they really rely on their host instead for that protection, instead of having a cell wall. And you see this type of kind of reliance on host. So the couple of organisms that actually rely on their host for plant interactions, like our rhizobium that we use in our lab, that will have a diminished cell wall as well once they actually have that symbiotic relationship with plants. Okay. Even though in their free living form in the soil, they do have a cell wall. And so what happens is, if you don't have a cell wall, you can have this pleomorphic arrangement within your cell, in that the plasma membrane just kind of takes on whatever shape it takes on. And that is actually refers back to the name mycoplasma, because they just essentially have plasma instead of having a cell wall. So that's that cell membrane or plasma membrane instead of having a cell wall on top. These are very small organisms, and as we said, they're usually parasites. So they're about 0.2 to 0.8 micrometers in length. Some of the, if not the smallest bacteria that we know of, and they don't have a cell wall. We said that's very unique to these organisms, and sometimes to organisms that take on different forms when they make symbiotic relationships. Another really unique type of cell wall is in organisms that are called mycobacterium. Mycoplasma, mycobacterium, they're very different in their structure, so don't get them confused, and we're going to talk more about them in chapter 11 as well. Mycobacterium, there's a species of mycobacterium that causes tuberculosis. The way that it does that is it actually has this extra additional reinforcement within its cell wall of what's called mycolic acid, which is a wax that really makes it really hardy. And it can actually live within the white blood cells that come to attack it when it infects a human host. And so it lives within those white blood cells and within the clusters that our body forms of white blood cells, that just continues to replicate. And in that way, it produces or it causes tuberculosis. Okay. And so these are technically gram-positive organisms, but they do actually share some characteristics with both gram-positives and gram-negatives. So the reason why they're called acid-fast here is because we can't actually use a gram stain on them, these mycobacterium that have mycolic acid in their cell wall. We can't use a gram stain on them because that mycolic acid, that wax, actually repels the stains within the gram stain. We have to use what's called an acid-fast stain. So sometimes they'll be referred to as acid-fast bacteria. And so what you can see here, even though we classify them as gram-positives, and they tend to have relation or phylogenetic kind of connections to gram-positives, they have characteristics that actually match kind of both of these organisms. So they have a thin peptidoglycan cell wall, but their mycolic acid cell wall, or part of their cell wall, is added to that and is very thick. And so these two components look like the thickness of a gram-positive, even though technically only a small portion of that, just like a gram-negative, is actually the peptidoglycan cell wall. And so here you can see, as we saw before, how they're kind of infecting a single white blood cell and then an aggregate of white blood cells. And then your body essentially tries to like zone off that region. And then you actually get these kind of where they're reproducing within those kind of regions within the lungs. Okay, so far we've talked about these external structures to the cell. We've talked about the glycocalyces. We've talked about different kind of extrusions, so flagella and fimbriae and pili that come out of the cell. And then we've talked about the cell wall, which protects the cell, even though it's connected to and it's a component of the cell. The cell membrane is the first component that is kind of a required piece of all living systems. So even these exceptions like mycoplasma have plasma membranes, right? All cells are going to have plasma membranes. And you're probably familiar with plasma membranes from your previous classes. And so you have a phospholipid bilayer with various types of proteins that move through that, and oftentimes extensions above that allow for the cell to maintain a separation between it and its environment and interact with the environment and other cells as well, and then allows for flow of nutrients in and out in a selective way. Okay. And so you can see here, bacteria have a phospholipid bilayer similar to eukarya. When we talk about archaea, we'll see that there's a little bit of a distinction with archaea. And so you have these hydrophilic heads on the outside of the cell and the inside of the cell, and hydrophobic tails in the middle here. Now, this model that I have here is actually a eukaryote, but imagine that that plasma membrane is covering the whole cell, and it's this whole turquoise kind of area around. And of course, the prokaryotic cell wouldn't have those compartments within. And then if we were to zoom in, I have another model here. Both of these are art integration projects from previous classes, where you can see those hydrophilic heads covering the whole surface of the cell and also the inside of the cell. And these hydrophobic tails allow for that kind of internal layer within the plasma membrane. And then you have things like transmembrane proteins or different types of lipids and sugars that are on the external of that cell membrane. Okay. And so what I want to do is, this is actually the end of part 1 of this chapter. And I'm going to have you pause and watch 2 or 3 videos, up to you. I have a video that takes you through the fluid mosaic model of the plasma membrane. It's a little bit antique, but it's my favorite still. Then a review of the functions of the plasma membrane and passive and active transport. Okay, that's the second one. And that's kind of generic to plasma membranes generally, throughout all domains, generally, with some exceptions. And then there's a really detailed video by Dr. Bollock, if you want to go into more detail about gram-positives, gram-negatives, acid-fast bacteria, and mycoplasma, where really he gets into details about how those distinctions kind of are and how they impact the function of the cell wall and the cell membrane. And just a terminology point, cell wall and cell membrane together, we call them the cellular envelope oftentimes, but the cell membrane is directly up against the cytoplasm, and then the cell wall is above that. Okay, so let's take a quick movie break. We'll pause the lecture here, take a break, come in, watch either 2 or 3 of those videos that are linked below, depending on how much detail you want to get into, and then come back in, and we're going to continue our discussion of bacterial anatomy. Okay, see you in a bit.